after all, even living through this pandemic, we have had success at our institute and at our Center of Excellence. So this year's annual report for 2020 will be very, very special. You will have a lot of movies and interviews from our institute. Please enjoy 2020 from our Center of Excellence and Kavli Institute.
The Kavli Institute is an interdisciplinary village of neuroscientists, all specialized in their fields, but having in common the desire to understand how complex information is encoded in high-level neural networks and systems.
The core activity at our institute is following our vision, which is that we try to understand how the brain is generating higher mental functions. And the researchers working at our institute, they come from diverse background, which makes this a very, very exciting place to be, to address different questions.
So in this annual report, we want to highlight 2 research publications from the previous year.
The first one is from Emre Yaks's group. He is studying tiny baby zebrafish, and still he can understand processes in the brain that we have dreamt about for a long time. What he has been doing is to follow cells from early birth and where they end up in the brain. And he is seeing then that cells that are born at the same time have same functions. So this is the first step now for our institute to dig into these questions regarding development. So this is so fun. Enjoy.
It's important to understand life and how life works and how brain works. How all these things put together, which eventually lead to our minds and our brains. We are using a combination of microscopy, physiology, and animal behavior to study and understand how the sensory information is interacting with the internal dynamics of the brain and how these interactions lead to behavioral plasticity. So we are interested in these ancestral corticolimbic structures in zebrafish brain that correspond to our hippocampal and triangle-like areas. And we want to understand the universal circuit architecture and computations that underlie how these brain regions leads to learning and they provide us this behavioral plasticity. My lab have been working on habenula. This is a brain region that is important for predicting outcomes, which, as you can imagine, important for both learning but also for decision making. As animals develop, habenula also develop, but in a way where distinct functional modules of the circuit is added sequentially. For example, one module is born at a certain developmental stage and the next module born, and so on and so on. Interestingly, in a separate paper, we also show that actually as animal develops, they actually gain new abilities in their learning. For example, a really young zebrafish are not very good in learning, but as they develop, they actually get better and better in learning. Actually, when we perturb distinct modules of this circuit, we also find out that not only the learning can be perturbed, but also relearning can be perturbed. In a way, for example, animals learn one rule, but then during the experiment, we change the rules of the game, so they have to learn a new rule. And actually perturbing that particular nucleus in habenula lead to actually an impairment in this reversal learning, which was also, I think, at least to me, it was very exciting.
And I think this is important for the Kavli Institute as a whole, because we know that by studying development, we also learn how the brain works together, how it's assembled, and I think it provides many clues to the workings of the adult brain.
The second story that we would like to present to you is from Jasse Rudi's group. His vision is to use his ideas and theories and computational tools that he has from physics and bring them out on other questions. And he said that I can describe the universe with these models and calculations and population of people, and he is willing and interested in using it to explain brain processes. He has worked on a very important question. How do we remember, and how does the brain work?
The Rudi group have shown what conditions need to be present for maximizing information storage or capacity in brain networks. They have shown that by using plasticity, as we call it, or different tunings of connections between brain cells, you can actually store information in a very efficient way, much better than, for example, what you get in traditional, more theoretically funded, not so realistic models of how the brain might work.
How do you learn stuff? How effective is your brain at storing memories? Who is the more efficient learner, the mammal brain or an artificial intelligence? This is a short story about the fascinating findings of the brain's capacity for memory by a small group of theoretical neuroscience researchers at the Kavli Institute for Systems Neuroscience in Trondheim and CISA School in Italy. So how does the brain learn? The brain builds your memories in tissue. Tiny connections called synapses form between neurons that are attentive to similar tasks. The synapses allow neurons to join forces in networks so they can collaborate with other specialist neurons to generate high-level brain functions, such as learning and memory. As you learn new stuff and recall old memories, your synaptic connections are continuously being molded to support your new knowledge. In the brain, the magic happens locally. It is self-organized by pairs and pairs and pairs of synaptically connected neurons, collaborating to code your experiences, feelings, thoughts, and actions in the brain's own language. So, is this similar to how artificial intelligence learns stuff? Actually, no. Robots and computers learn by an algorithm known as backpropagation. The algorithm is repeating over and over. Each cycle reduces the number of errors in the network. Since the 1980s and the rise of the algorithms for artificial learning, the general opinion has been that the artificial intelligence learning rules are more efficient than that of the brain, reinforced by historic milestones in deep learning, like in 1997 when the computer Deep Blue defeated Kasparov, the world's best chess player. But could this be the whole truth? Scientists at CISA in Trieste and the Kavli Institute for Systems Neuroscience in Trondheim pondered over some assumptions in a body of research supporting this opinion that may not be entirely correct. First, the brain is not a binary network. Second, the AI learning rules are centrally supervised, which means they're slow. The computer needs to collect a lot of information from the whole network for each cycle, while the brain, on the other hand, distributes the tasks across its network of locally self-organized neurons. By using Gardner's storage capacity on a biologically plausible model of how the brain operates, the scientists found that the brain's learning rule can be just as efficient, if not more efficient, than that of artificial learning. So even though AI may defeat the brain in a game of chess, when it comes to storing memories, the brain wins.
And this is also extremely important for the Institute because it shows the importance of theory in guiding our experiments, and also it addresses one of the most fundamental questions of how the brain works. The starting point of our investigation is our ability to understand space, time, and memory, high-level brain functions that we share with all mammals. These abilities are generated in brain regions called the medial entorhinal cortex, the lateral entorhinal cortex, and the hippocampus. Brain functions such as space, time, and memory start to offer a window, one of the very first of its kind, into the most complex processes within our brains. However, these functions in entorhinal cortex are also important because they are among the very first ones to be disrupted in Alzheimer's disease.
We started out with the grid cells that we discovered in 2005, and we have expanded this to different functional cells, and these cells are found in entorhinal cortex.
We discovered a new type of principal neuron called the object vector cell that is fundamental to our brain's navigation system. While grid cells map and measure open space, object vector cells measure space relative to objects and landmarks that occupy it.
And when such cells die, then people are typically diagnosed with Alzheimer's disease, and they lose functions like finding their way in the environment.
Since the 1950s, neuroscientists have been able to record from single neurons, revealing how individual neurons pick up on specific properties of the external world. While such recordings have paved the way for modern neuroscience, they still fall short when it comes to explaining how brain functions arise as the result of interactions in neural circuits and neural populations. Our higher brain functions are generated in large neural networks that are widespread across the brains. To understand how these networks operate, we must monitor activity from thousands of neurons at the same time while the animal engages in experimentally controlled behaviors. Two new complementary techniques adopted and developed in our lab paved the way for such studies.
Two new tools that are just amazing. One, this huge microscope here, 500 kilos. This is a 2-photon microscope, and in this we can make a window into the brain and we can see the activity of the cells while the animal is performing. But of course, the animal can't run around with such a huge and heavy microscope on its head. So then, luckily, engineers have been able to develop a tiny 2-photon microscope. And this is so tiny, 2 grams, so that the animal can run around, even a mouse can run around with this microscope on its head, chasing chocolate, and then we can see all these beautiful cells that we have been focusing on, like grid cells, border cells, object vector cells, and all these cells. Because we have so many cells at the same time, we can then start to understand how the brain is generating such functions.
Second, we are using new miniature silicone probes that allow us to listen in on the conversations at very high temporal resolution that take place between hundreds and thousands of neurons while animals are running around freely in the environment.
We have a shank and it has a lot of sensors. So when we stick that into the brain and listen to the electrical activity in the brain, we can suddenly record thousands of cells at the same time. And you know, when you can do that, then you can start to talk about how populations and big numbers of cells generate our mental functions.
With the emergence of new technologies, we are today on the verge of understanding not only the words, but also the sentences in the neural chatter that generate our higher cognitive functions. Specifically, we are on the track of understanding how the grid pattern arises, and this will tell us a lot about how the cortex encodes information in a general sense and how patterns arise in this part of the brain. This is a totally new way of understanding the brain, and this is the future. So in 2020, we received a synergy grant from the European Research Council, which was a collaboration between our Kavli Institute and the Hebrew University in Jerusalem. This is a major research grant which allows us to merge theoretical neuroscience with experimental neuroscience. So we want to use theory to guide our experiments to understand one of the most important cognitive functions, namely spatial navigation, or how we find our way in the environment. One of the greatest mysteries in human inquiry is how activity in brain cells or neurons give rise to the broad spectrum of high-level brain functions, such as thinking, planning, remembering, imagining, and so on. There's a lot of evidence suggesting that these brain functions must arise in interactions among thousands of interconnected neurons in the brain. But we haven't really been able to measure these interactions in any way so far. With the recordings from single cells, we have been able to uncover the elements of the space circuit, such as grid cells, but we haven't been able to understand how they work together. Now there is a revolution going on in the field of neuroscience, because it is suddenly have become possible to record activity from many thousands of neurons with known connectivity at the same time. And in our lab, we are introducing two state-of-the-art tools. First, NeuroPixels probes that allow us to record the electrical activity of thousands of neurons. And second, 2-photon miniscopes that can be carried on a mouse's head and allow us to visualize the activity in the brain as a mouse is solving a cognitive task. In order to understand brain computation, experiments are not enough. We need to also develop and test theoretical ideas or models of how computation arises. Yoram Burak is a computational neuroscientist and a theoretical physicist who has developed models of how spatial firing patterns arise in large populations of interconnected neurons.
My research team and I develop theories for how neural networks in the brain operate and compute. We think that there is now an unprecedented opportunity to examine the validity of such theories at a very detailed and mechanistic level. By designing experiments together with the Moser Lab for this purpose, in which hundreds and thousands of neurons will be simultaneously monitored and perturbed, we will test for the first time existing classical theories of neural computation in the brain, and we also expect to greatly refine our theoretical understanding.
Our goal is to pioneer the investigation of one cognitive function, spatial orientation, that we believe have all elements in place for a breakthrough mechanistic understanding to take place. The project will pave the way for a better understanding of how the brain produces high-level cognitive functions. This will in turn allow us to understand the causes of diseases where these high-level functions are compromised, such as in neurological diseases and psychiatric syndromes. The ERC Synergy grant represents a total transformation. I think this change of activity benefits not only our own research group, which heads this grant, but also the entire institute. So for the next decade, the institute is going this way. We are trying to understand how huge neural networks of hundreds to thousands of cells work together to create the most advanced functions in the cortex of the brain. NorBrain 3 is a large-scale research council infrastructure investment that gives Norwegian researchers access to these new technologies. In NorBrain 3, we shall both develop and make available these technologies for next-generation systems neuroscience. Our Kavli Bråten Center for Cortical Microcircuits is a legacy to humanitarian and philanthropist Mrs. Pauline Bråten's support to foundational science for Alzheimer's disease. We made a great effort to build on her initiative with the aim of establishing an Alzheimer research center. And thanks to generous support from the foundation Stiftelsen Christian Gerhard Jepsen, we are now establishing the K.G. Jepsen Center for Alzheimer's disease, in which we shall link our work on animal models to studies of human subjects, including at-risk Alzheimer patients, in tasks similar to those that we use for investigating mechanisms of space, time, and memory in rats and mice.
We are trying to understand how the brain is working, and that is us. Who would we be without our memories, without finding our way? it's important for us as humans to start to understand what is happening inside, but also to understand what happens when it goes wrong, when cells die and our brain doesn't function like we would like it to do. So that is why I think our research is just a game changer.
I hope you enjoyed this annual report. Now at the very end, we would like to thank all those who made this work possible. The Research Council of Norway, the Kavli Foundation, the European Research Council, the Foundation Stiftelsen Christian Gerhard Jepsen, Pauline Broughton and her family, and the ideal organization Ensliges Landsforbund, who in 2020 made a substantial contribution to the Alzheimer's disease-related work at the Institute. Thank you to everyone.